How are you everyone? And today we are going to talk about the concepts of enzymes. Before going to enzymes, we are going to talk about proteins. So proteins, or the function of proteins, is simply one is for immunity such as antibodies. Another function of protein is for hormones or growth hormones, for example. Another one is transport, and that would be an example to this is hemoglobin. And another one is for structure, that is given by the example of collagen. And eventually, one of the functions of protein is for catalysis, and that one will be about enzyme, and this is our topic for today. So what are the important functions of enzymes? Enzymes are actually part of the catabolic reaction, such as cellular respiration with ATP. Another one is enzyme is also part of the function of our body's detoxification process that is done in our liver. And enzymes can be found also in the process of digestion in our digestive system. And in terms of anabolic reaction, enzyme is also a component of that one, such as in the photosynthesis for plants and also for protein synthesis in animals. So basically, enzyme lowers what we call the activation energy barriers. And what is that? So in a regular scenario without the use of enzyme, this is the energy requirement for a biological process to occur before the production actually of that process happens. But with the use of enzyme, that is lower down. So basically we do not need much of the energy for the different bodily process, because enzyme will help us lower down the activation energy barrier. And because of that, the activation energy barrier that is lowered will help speed up the rate of reaction of the different biological processes. When we talk about an enzyme, so basically how does an enzyme looks like? Well, an enzyme has actually what we call an active site. So that is the entire enzyme, and the active site is the one on top of the entire enzyme. So this is the entire enzyme, and on top is what we call the active site. And another one is that this enzyme will be acting on the substrate. So basically the substrate are actually biological molecules that will undergo the different processes. So by the help of an enzyme, this biological molecule, or what we call a substrate, will be undergoing those processes. But how does this work is that the substrate also has a complementary shape that will actually complements to the active site of an enzyme, and with that this substrate can now bind into the active site of the enzyme to form what we call the enzyme substrate complex. So what is an enzyme substrate complex? Actually, this is now the coming together of the substrate and the active site of the enzyme. So basically they will form as one material later. However, the enzyme will not be destroyed in this process, and it will only be used to process the substrate to form the different products of the biological processes. And talking about the enzyme substrate complex, there are two hypothesis models. The first model is what we call the lock and key, and the second model is the induced fit. So going back to the enzyme hypothesis model, again, there are two models here. The first is lock and key model. And in the lock and key model, that our substrate is complementary to the active site shape. So basically it resembles the lock and the key. so that the substrate shape should complement also to the enzyme active site. And this type of idea, or this idea, is actually first postulated by the German scientist named Emil Fischer in 1894. So the other type of enzymatic hypothesis model is called the induced fit. And with this is that when there is a substrate, there will be an induction or the forcing of the uh, active site of our enzyme to fit into the shape of our substrate. And so that is suggested by the scientist named Daniel Koshland in 1958. So whatever the model that the scientist is actually looking into, or they prefer into, the same basic principle is working here. So basically, the working principle of an enzyme is that it just fits into the substrate, and with that, it can already starts to work or function as to what biological process that it will do. So going back into the different enzymatic actions again, so our enzyme works as a kind of biological help to lower down the actually the energy barrier for our different biological process to work. And there are sample enzymatic actions here. First is we have the enzyme called catalase. This actually catalase will have a substrate called hydrogen peroxide, and the product is actually producing oxygen gas and water. So another one is diastase. So the substrate is a starch and it will produce maltose. Well, another one is a pectinase, and the substrate is pectin in plant cell walls, and it will produce simple sugars. So it's actually releasing of juices from the cell. And we have pepsin, very popular type of enzyme that works on the substrate called protein, and it will produce the short polypeptides. And also another example here is called the renin, and the substrate is actually the milk protein that is called the soluble casein, and the product will be the insoluble casein, which is the curdled milk. Enzyme. It is a biological catalyst that speeds up the rate of reaction. This enzyme lowers the activation energy barriers that speeds up the rate of reaction. The rate of reaction, however, is affected by what we call the limiting factor, and these limiting factors are the following. Enzyme concentration. This enzyme concentration, when increased, will further increase the enzymatic activity. However, when it reaches the saturation point, that enzymatic activity lowers down. Another one is substrate concentration. This is a kind of limiting factor that affects the rate of the reaction or the enzymatic activity directly. And another one is when it is outside of the optimum value. The optimum value of pH of an enzyme. So an enzyme has a optimum operating pH levels, and another one is the optimum operating temperature of an enzyme. So basically, this is the reason why one is asked, or it is much encouraged to drink lukewarm water after eating, because if you can see, it can affect our digestive enzymes. So digestive enzymes are actually working in an optimum value between 30 degrees centigrade up to more than 50 degrees centigrade. So lukewarm water can help our digestive enzymes. Now, the concept of the optimum value of an enzyme actually maximizes enzymatic activity. So these are temperature and pH. When you speak of temperature, that is if the environment condition of an enzyme is operating at a very low temperature, enzymes are inactive, and if the condition is very high, for example, the environment of an enzyme is having a very high temperature, and that will cause the enzyme to denaturize. So denaturization of an enzyme is actually an irreversible process, meaning to say that enzyme will already be decomposed back to its primary structure of the protein, which is the primary structure of, or the chain of amino acids. And another one is pH. When pH is too high or pH is too low, the same thing happens. Enzyme will denaturize. So what are the six types of enzymes? We have the group of oxidoreductases. Another group is transferases. Another one is hydrolases, the lyases, isomerases, and finally ligases. So the first group of enzyme is actually a hydrolase enzyme. The examples of this type of enzyme are lipase and protease. This group of enzymes actually is going to handle the catabolic reaction or hydrolysis. So we have an example here, a triglyceride or triglycerides with the use of lipase enzyme is going to hydrolyze this into glycerol and fatty acids. So that lipase there is an example of a hydrolase enzyme. So to continue with the hydrolase, we are now going to look at how protease, an enzyme of a hydrolase group of enzyme, is going to hydrolyze or break down our protein molecule. By the means of the process of digestion, protease is actually utilized in breaking down of protein molecule into subgroups such as the different amino acid molecules. So another type of enzyme or enzyme group is actually called isomerase. An example to this one is the phosphohexoisomerase. And this type of enzyme actually arrange atoms within a molecule. So we have an example here. Glucose 6-phosphate can now be rearranged into a molecule called fructose 6-phosphate. Or likewise, since it is a reversible process, fructose 6-phosphate can be rearranged into glucose 6-phosphate by the use of the enzyme called phosphohexoisomerase, a type of isomerase enzyme. So another group of enzymes is the lyasase group. So the lyase enzyme, and the examples given here are the carboxylases and the other one is aldolases enzyme. And the function of lyase enzyme is simply splitting chemicals into smaller parts without the use of water. So an example here is in the glycosidic reaction, the fructose 1 and 6-biphosphate is actually broken down or split into glyceraldehyde 3-phosphate, and another one is dihydroxyacetone phosphate. So again, the aldolase enzyme is the type of enzyme that is used in the splitting of these chemicals into smaller parts. So to continue, this time we are going to make use of the reaction of pyruvate to produce carbon dioxide and acetaldehyde. Another type of lyase enzyme is called decarboxylase enzyme or pyruvate decarboxylase enzyme. So it is this kind of reaction that splits the two chemical components. One is carbon dioxide and acetaldehyde from a pyruvate. So that is an example of this type of enzyme. And so we have another type of enzyme or another group of enzyme classification, the oxidoreductase enzyme group, and in our example, we are making use of dehydrogenases and oxidases group of enzymes or types of enzymes that belong to oxidoreductase. And what does an oxidoreductase do is that it will transfer electron or a hydrogen atom from one molecule to another. And for this type of example, we are going to make use of malate, a molecule of a biological molecule, and we have the NAD. By the use of the malate dehydrogenase, we are going to transfer the hydrogen from malate into the NAD, so that it will now become NADH plus H. And so with that, the malate can now be converted into oxaloacetate. So to continue with the oxidoreductase classification of enzyme, we are now going to make use of oxidase, a type of this oxidoreductase enzyme. And we'll see how oxidase transfers an electron or hydrogen atom from one molecule to another. And in this example, we are going to make use of beta-D glucose. Then we have the oxygen. With the use of glucose oxidase enzyme, we are going to transfer that hydrogen into the oxygen, and with that we are going to produce a D-glucono 1 and 5 lactone and hydrogen peroxide as the product of the catalytic reaction. And so another type of enzyme or another enzyme group is called synthetases, and the examples here are DNA ligase and DNA polymerase. So in a synthetases type of enzyme group, they are actually going to join two molecules by the formation of new bonds. This is happening in a anabolic type of biological reaction. For example, if we have the DNA backbone, which is the red one in here with a sticky end, and the insert DNA with another sticky end, so by the means of this enzyme, in the process of ligation, the two strands are now attached together, and so this method is called the recombinant DNA. So recombinant DNA actually utilizes ligase type of enzyme, which is a which belongs to a group of synthetase enzyme. So the enzyme group called transferase are actually given by two examples in here, and that is kinases and transaminase. So this type of enzyme group actually moves a functional group from one molecule to another. As an example, we have the molecule of creatine, and with the ATP, our creatine kinase will remove that phosphate group from that ATP to be transferred to our creatine to create the phosphocreatine molecule. Now our enzyme active sites are actually affected by a molecule that binds to the enzyme. And this molecule, they are known as the enzyme inhibitors. And so these inhibitors can actually prevent the enzymatic activity, or if not, they slow down the speed of the enzymatic reaction. So enzyme inhibition is caused by, again, the enzyme inhibitors, and they are either organic or inorganic origin. And enzyme inhibition, the importance to this one is for the regulation of our enzyme activity, as there are biological processes that will already signals when to stop the biological process, such as in the conversion of energy or in the glycosidic processes. And another one is the accumulation of intermediates in the cellular metabolic pathway. So again, this is when the intermediates are already accumulated, so it can also be inhibited by the enzyme inhibition. And another one is identification of the catalytic functional group. So the inhibition is also made use in this type of process. And also in the development of a drug for therapy, this is either the making use of the competitive inhibitor as a type of drug, or a suicidal inhibitor as another type of drug. Moving on to the enzyme inhibitors, there are several types of enzyme inhibitors, and one is allosteric. This type of enzyme inhibition is actually enzyme has what we call the allosteric site. The function to this one is actually to increase or decrease the enzymatic activity. And another type of inhibition is the irreversible. The irreversible inhibition is also known as the suicide inhibition or suicide inhibitor, because the inhibitor will bind to the enzyme active site and will actually denaturize the enzyme permanently, so therefore the enzyme can no longer function. And the other type is also the what we call the reversible, meaning to say that these inhibitors, when they are removed, the enzyme can resume back to its natural enzymatic function. And the types are non-competitive inhibition. This non-competitive inhibition will actually bind at another site of the enzyme, and then it increase the substrate concentration. So similar to allosteric type of inhibition, they work almost the same, but this one will produce a reaction that will slow down. Okay, so the reaction of the enzyme will actually slow down. And then another one is the competitive inhibition, wherein that the inhibitor will compete with the substrate for binding. So the substrate can no longer bind into our enzyme, because it will be competed with the inhibitor that it also inserts into the binding site of our enzyme. And the last one for the reversible type of enzyme inhibition is called the uncompetitive inhibition, that wherein the inhibitor binds to the enzyme substrate complex. That is uh, very similar to non-competitive, but it's very close to the enzyme and a substrate complex, and that causes structural distortion to the enzyme, and it will also alter the function of the enzyme. So we are going to present some examples of this one later in the next slide. Allosteric enzyme inhibition. The inhibitor of this allosteric enzyme inhibition is actually going to change the composition of our enzyme. It will actually create two conditions, wherein when the inhibitor is already attached to the enzyme, it will create a deactivated enzyme, and when it is removed, it will create an active enzyme. So this is how the allosteric enzyme condition works. So in this case, this is our enzyme and this is the substrate, wherein the substrate is not allowed to be processed further when the inhibitor is already attached to the allosteric site. And when the inhibitor is removed from the allosteric site, so now the enzyme is in the active form. So we have the example, the ATP and citrate concentration when they are already in the actual concentration, such as saturation. These are actually a form of regulation to the enzyme phosphofructokinase 1. So in a glycosidic reaction, so when that is reached, so therefore the phosphofructokinase is already deactivated in this case. The irreversible or suicide inhibition is a kind of inhibition wherein the inhibitor binds to the enzyme active site and denaturalizes the enzyme permanently. So this is an illustration for that, wherein the inhibitor reacts with the amino acid in the active site and permanently denaturalizes the enzyme. An example to this, or the importance to this, will be in the manufacture of drugs that is used to prevent the proliferation of rapidly or fast growing cancer tumors, like preventing the thymine to create more DNA to produce those tumor cells. So this is one important aspect for understanding irreversible or suicide inhibition. So for the non-competitive inhibition, the inhibitor will bind at the site of the enzyme in another site aside from the active site. Then it increases the substrate concentration. So with this, it does not permanently stop the activity of the enzyme. However, it will only produce a slowdown reaction. So this is a kind of illustration that presents the how the non-competitive inhibitor works. And an example is actually the cyanide poisoning. This prevents our body cell from using oxygen and that the cell will suffocate and die. For the competitive inhibition, this is where the inhibitor competes with the substrate for binding. So in the case of this illustration, we can see that the inhibitor is having the same shape of our substrate. So when the inhibitor comes first to bind with our enzyme, then therefore this will be now creating the enzyme and inhibitor complex. In this case, there is no reaction. So in other words, that our substrate is blocked by the what we call the inhibitor. And an example to this one is our statin drug. So statin drug act as an enzyme inhibitor that binds into the active site of our enzyme, so that this will help regulate the production of cholesterol in the body, which in turn will make the person sick or ill. And this one is called the uncompetitive inhibition, that the inhibitor will bind to the enzyme substrate complex and will cause the structural distortion of the active site of the enzyme. So this one is that it requires already the enzyme to attach to the substrate, and when that happens, an uncompetitive inhibitor will also now be attached to the same closer to the substrate and enzyme complex. And so for example is alkaline phosphatases and enzyme are overexpressed in certain types of cancers. So with that, some amino acid residues act as an uncompetitive inhibitor, and this will regulate the alkaline phosphatases activity to further produce and forms cancer cells.